Bismillahirrahmanirrahim. Assalamu alaikum students. Today I will teach you about the molecular genetics, about the DNA, the genetic material. 20th century biologists realized that a molecule that serves as the genetic material must have certain characteristics to explain the properties of life. First, the genetic material must be able to code for the sequence of the amino acids in proteins and control protein synthesis. Second important point is that it must be able to replicate itself prior to the cell division. And third, the genetic material must be in the nucleus of the eukaryotic cells. And fourth, it must be able to change over time. Students, now I will tell you about the double helix model of DNA. Two kinds of molecules participate in the protein synthesis, DNA and RNA. Okay, DNA is the abbreviation of deoxyribonucleic acid and RNA is the abbreviation of ribonucleic acid. DNA is the genetic material and the other ribonucleic acids or RNA, it is produced in the nucleus and moves to the cytoplasm where it participates in the process of protein synthesis. The study of how the information stored in DNA codes for RNA and protein is called as molecular genetics. DNA and RNA, these are larger molecules which are made up of important subunits which are called as nucleotides. Okay, a nucleotide consists of a nitrogen containing organic base either in the form of double or single. A nucleotide structure. A nucleotide basically consists of nitrogenous containing organic base, a double ring which is known as purine, and a single ring which are known as pyrimidine. Okay, nucleotides also contain a sugar, 5-carbon sugar, which is called as pentose sugar, and a phosphate group. Both DNA and RNA actually having some differences. Okay, both these are having purine bases, adenine and guanine, and the pyrimidine base cytosine. The second pyrimidine in the DNA is thymine, whereas in RNA it is uracil. This is important difference between the first important difference between DNA and RNA. The second difference between DNA and RNA involves the sugar. Okay, because in DNA there is deoxyribose sugar, whereas in RNA it is ribose sugar. Third important difference is that DNA is a double-stranded molecule, whereas RNA is molecule. Now I will tell you something about three-dimensional structure of DNA. The DNA molecule is ladder-like with the rails of the ladder consisting of alternating sugar phosphate groups. The phosphate of a nucleotide attaches at the 5th carbon of the deoxyribose. Adjacent nucleotides attach to one another by a covalent bond. Remember this point that the adjacent nucleotides, these are attached to one another by a covalent bond between the phosphate of one nucleotide and the 3rd carbon of deoxyribose. Similarly, the pairing of nitrogenous bases between strands holds the two strands together. Adenine is hydrogen bonded to its complement thymine, okay, whereas guanine always pair up with hydrogen bond to its complement cytosine. Each strand of DNA is oriented such that the 3' carbons of deoxyribose in one strand, these are oriented in the opposite directions from the 3' carbons in other strand. The strands terminal phosphates, these are at opposite ends. DNA molecule is thus said to be anti-parallel. Okay, this is very important point, students. The entire molecule is twisted into a right-handed helix with one complete spiral of every 10 base pairs. Okay, this is the double helix model of DNA, and these are important points of double helix structure of DNA. And this is the diagram showing the replication of DNA. this is the DNA the important enzyme which is known as DNA polymerase, okay, which actually initiates the process of DNA replication. And this is the initiation site, okay. And another important enzyme which is required for the DNA replication is DNA ligase, okay, which actually join together the fragments of the DNA. DNA replication in eukaryotes. What's happened during the DNA replication, that each DNA strand becomes the template for a new strand. It means the pairing requirements between the purine and pyrimidine base dictate the positioning of the nucleotides in a new strand figure, okay. Thus each new DNA molecule contains one strand from the old DNA molecule and one newly synthesized strand. Because half of the old molecule is conserved in during the DNA replication, that's why the process of DNA replication is said to be semi-conservative. Now we'll study about the genes in action, how the protein synthesis process takes place. A gene can be defined as a sequence of the bases in DNA that codes for the synthesis of one polypeptide chain. Genes actually transmit the information from the nucleus to the cytoplasm where protein synthesis takes place. The synthesis of an RNA molecule from DNA is called as transcription, which is actually the first step in towards the process of the protein synthesis. And the formation of a protein from RNA at the ribosome unit is called as translation process, which is the second step in the protein synthesis. Now students, I will tell you that there are three major kinds of RNA which are actually involved in the process of protein synthesis. Each RNA has a specific role in protein synthesis and is produced in the nucleus from DNA. The first important category is messenger RNA, which is abbreviated as small m RNA. It is linear strand that carries a set of genetic instructions for synthesizing proteins to the cytoplasm. Okay, and the second important type is transfer RNA, which picks up amino acids, actually specific amino acids in the cytoplasm, which is actually required to synthesize the specific polypeptide chain. Okay. And the third important category of RNA is ribosomal RNA. Ribosomal RNA along with proteins, it actually makes up ribosome. Okay. What is genetic code? The DNA basically code for 20 different types of the amino acids found in all organisms. The information carrying capabilities of DNA reside in the sequence of nitrogenous bases. Do you understand, students? The information carrying capabilities of DNA, it basically reside in the sequence of nitrogenous bases. It means that sequence of nitrogenous bases play a very important role. What is a genetic code? Genetic code is a sequence of three bases which are also called as a triplet code. Okay. Each three base combination is a codon. More than one codon can specify the same amino acid because there are 64 possible codons. Okay, how many codons exist? 64 possible codons exist, but only there are present 20 different types of the amino acids. Amino acids are, students, these are building blocks of proteins. Okay, and there are present thousands of different types of the proteins, but the building block of protein are amino acids, which are only 20 in nature. It means that there are present different combinations of the amino acids which are specific for specific polypeptide chain. This characteristics of the code is referred to as degeneracy. There are present two important codes, initiation codes and termination codes. Okay. The base sequence AUG, it codes for the amino acid methionine, which is a start signal. The base sequence UAA, UAG and UGA, these are all stop or termination signals that actually indicate where polypeptide synthesis should Now we'll study about the first step of the protein synthesis, which is transcription. Okay. In transcription process, the genetic information resides in the DNA is first transcribed into the messenger RNA. Okay. Double-stranded, the information which is actually reside in double-stranded DNA, it is transcribed into single-stranded messenger RNA, you can say in simple words. Transcription involves numerous enzymes that actually play a very important role to unwind the region of the DNA molecule, to initiate and end messenger RNA synthesis, and similarly to modify the messenger RNA after process of transcription is completed. Okay, so whole of these processes actually require different enzymes. Unlike DNA replication, only one or a few genes these are exposed, and only one of the two DNA strands is transcribed. Remember this point, students. In replication, both DNA strands these are actually involved in the process of replication to synthesize the two DNA molecule. Okay, but during the transcription process, only one of the two DNA strand is only transcribed. Okay. One of the important enzymes of this process is RNA polymerase. After a section of DNA is unwound, the RNA polymerase recognizes a specific sequence of the DNA nucleotides. This is important function of DNA nucleotide to read the genetic information which is present in the DNA. Okay. The DNA polymerase attaches and begins joining ribose nucleotides which are complementary to the 3' end of the DNA strand. The newly transcribed messenger RNA is called the primary transcript. It must be modified before leaving the nucleus to carry out protein synthesis process. As you know, students, that protein synthesis machinery exists in the cytoplasm. Okay, so that's why when messenger RNA is actually formed, or the newly transcribed messenger RNA is actually formed in the nucleus, then it is actually moved towards the cytoplasm where protein synthesis machinery is present. And what is protein synthesis machinery? It is ribosomes. Okay, these are ribosomes. Some base sequences in newly transcribed messenger RNA, these don't code for the protein. Okay, and thus there is actually requirement to cut down these non-coding regions. Okay, and this process is called as RNA splicing, which involves cutting out non-coding regions so that the messenger RNA coding regions can be read continuously at the ribosomes. Okay. Second step of the protein synthesis is translation. Now, students, you should know the definition of translation process. Translation is the protein synthesis process at the ribosomes in the cytoplasm. Okay, based on the genetic information, the transcribed messenger RNA. Okay, why this process is called as translation? This is very important to know. Okay, because the the language of the genetic information which is actually present in the DNA is in the form of nucleotides. Okay, students, whereas the language of the genetic information which is actually present in the single-stranded messenger RNA is also in the form of nucleotides. Okay, whereas newly synthesized polypeptide chain or the protein is actually in the form of amino acids. As I had told you earlier that amino acids, these are building blocks of the proteins. So this is the process where the language of the language of genetic information which is present in single-stranded messenger RNA is actually translated into the amino acid language of proteins. Okay, so that's why here language is changed from nucleotides to amino acids. That's why this process is given the name translation. Okay. Important transfer RNA play very important role in the process of the translation. Okay, it brings different amino acids into alignments coded for by the messenger RNA to make the specific chain, And during translation, pairing of the messenger RNA codon with its complementary anticodon of transfer RNA appropriately positions the amino acids that transfer RNA. This is the diagram of clover leaf-like structure of transfer RNA, and this is the diagrammatic representation of the structure of the transfer RNA. An amino acid attaches to the 3' end of the molecule, which is indicated here in the form of arrow. The anticodon The anticodon is the sequence of 3 bases that actually pairs the codon in messenger RNA, thus positioning the amino acids that transfer RNA carries. Now we'll study about the ribosomes, which play a very important role in the process of translation, because ribosomes, these are protein synthesis machinery, and these are scattered in the cytoplasm. So that's why newly transcribed messenger RNA must leave the nucleus to get attached to the ribosomes. Ribosome is basically the site of the protein synthesis, which consists of 2 important units, smaller and larger units, that recognize the pairing between the codon and the anticodon regions. Several sites on the ribosome, these are binding sites for messenger RNA. At the initiation of translation, messenger RNA binds to a small separate ribosomal subunit. This is very important point, students. At the initiation of translation, messenger RNA first of all binds to a small separate ribosomal subunit. Attachment of the messenger RNA requires the initiation codon, which is AUG of the messenger RNA, be aligned at the P site of the ribosome. And where this P site is present, this P site is actually present in the smaller subunit of the ribosome, which is actually attached to the messenger RNA. The transfer RNA with the complementary anticodon for methionine binds to the messenger RNA first, Then a larger subunit of ribosome also joins, forming a complete ribosome. Got, students? Do you understand, students? And after this, when larger subunit of the ribosome also joins, then polypeptide formation is then begin. Another site, A site, is next to the P site. A second transfer RNA whose anticodon is complementary to the codon in the A site is then positioned just like this one. In B diagram, is actually indicating the 2 transfer RNA. These are aligned side by side in the B diagram. This step actually requires enzyme aid, and energy is also required for this process. And which is provided in the form of guanine triphosphate, which is abbreviated as GTP. An enzyme, peptidyltransferase, it is very important enzyme, students. Peptidyltransferase, a part of the largest ribosomal subunit, it breaks the bond between the amino acid and transfer RNA. This is very important enzyme, peptidyltransferase. And what is the function of this peptidyltransferase enzyme? That this enzyme is basically involved to break the bond between the amino acids and the transfer RNA in the P site, and catalyzes the formation of a peptide bond between that amino acid and the amino acid in. The messenger RNA strand then moves along the ribosome a distance of 1 codon. When it reaches 1 codon, then it moves along the ribosome a distance of 1 codon. Transfer RNA with 2 amino acids attached to it, it was in the A site, is now in the P site. Do you understand, students? A third transfer RNA can now enter the exposed A site, and this process then continues until the entire messenger RNA it has been translated. Translation ends when a termination codon is actually reached. And what is the termination codon? It is UAA.